Hello everyone, today we are going to discuss another laboratory technique, that is molecular cloning. So what is molecular cloning? Molecular cloning is production the production of the recombinant DNA molecule in a bacterial host. So in the molecular cloning, what happens? Recombinant DNA molecule is produced in the bacterial host. Now let us discuss about the steps of molecular cloning. There are total 5 steps of molecular cloning that you should know very well. So first, what we do, we isolate the eukaryotic messenger RNA of interest. We isolate the eukaryotic messenger RNA of interest. The messenger RNA, the YAM RNA, should be post RNA processing. Post RNA processing. So next step, what we do, we add the reverse transcriptase. We add the reverse transcriptase to produce the complementary DNA. Here the reverse transcriptase is the RNA dependent DNA polymerase. The reverse transcriptase is the RNA dependent DNA polymerase, and the complementary DNA here produced is lack of introns. There is no introns. There are no introns in the complementary DNA. Then what happens in the next steps? In the next step, what we do, the complementary DNA fragments are inserted into the bacterial plasmids. These complementary DNA fragments are inserted into the bacterial plasmids that contains the antibiotic resistant genes. Another step, after we insert the complementary DNA into the bacterial plasmids containing antibiotic resistant genes, there is insertion of the recombinant plasmid into the bacteria. Insertion of the recombinant plasmid in the bacteria. Another last step, what we do, the surviving bacteria that we have previously put in the antibiotic resistant gene, they produce the clone DNA. So the bacteria that survives on the antibiotic medium produces the clone DNA, that is the copies of the circular DNA. So this is the process of the molecular cloning. So we have discussed the definition of the molecular cloning and the 5 steps of the molecular cloning. Each have been described here. Thank you.